Should I phone him?
The broadcast is now starting. All attendees are in listen-only mode.
Hi, I just have a technical question. Do I press start broadcast or not? Okay, it's done. Okay, great. Yeah, yeah. So, okay, I don't need to press start broadcast. Am I on, Mark? Sorry about this. Yeah, let me know if not. Okay, well, hello everyone, and welcome to this second of 3 talks exploring the origins of the SARS coronavirus that is the cause of the current pandemic. For some, that means welcome back, and to those of you newly arrived, I hope to make this easy enough to follow that you won't be at too much of a disadvantage, even though these 3 talks are designed to be seen in chronological order. In the previous one, I suggested that the available evidence supported 2 broad conclusions. The first was that the major zoonotic origin theory initially shared in the media has essentially no evidence to support it. This theory was that the precursor to the SARS-CoV-2 virus, which for the purposes of this talk I'll be calling SARS-2, evolved inside a pangolin that was transported to Wuhan, possibly via the Huanan live animal market in the city. I also indicated that no credible alternative zoonotic origin theory had emerged to replace it. This absence of a zoonotic pathway is itself a surprising fact, since the 2 previous pandemic coronaviruses, SARS-1 in 2002 and MERS in 2012, were rapidly followed by the detection of their sources. Extremely closely related viruses were quickly found in animals to which the early victims have been exposed. Considering that sampling methods, sequencing technology, and epidemiological understanding are far ahead of where they were in those epidemics, the failure to find a credible zoonotic source ought to be puzzling. The second of these conclusions was that there is, on the other hand, plentiful evidence favoring a lab escape of some kind. There is the close proximity of a top-backed coronavirus laboratory, the Wuhan Institute of Virology, which I'll be calling the WIV, to the epicenter of the outbreak. There is the fact that the WIV collected SARS-related coronaviruses from bats and held in its freezers the nearest known relative of SARS-CoV-2, the current pandemic virus. In the last lecture, we discussed this near relative virus, which is called RATG13. Moreover, the WIV specialized in genetically manipulating coronaviruses from the exact viral species to which SARS-2 belongs. There are many coronavirus species, but what are we to suppose when the one species for which they had a particular focus happens also to be the one that is today causing the pandemic? There is the apparently pre-adapted quality of the virus, of the pandemic virus. From the very beginning of the outbreak, the SARS-2 virus was highly contagious and seemingly perfectly adapted to humans. SARS-2 has behaved all along as if it had not really recently jumped from another species at all. Coronavirus research at the Wuhan Institute of Virology is headed by Dr. Zhengli Shi. In this photograph of her, taken by Scientific American, Zhengli Shi is on a collecting trip to capture bats suspected of harboring coronaviruses. An important thing to notice about this picture is her protective equipment. Apart from some thin gloves, she isn't wearing any. This is a researcher who travels the world warning us all of the enormous dangers of human contact with bats, herself engaging in risky behaviors with bats. What are we to infer about how her laboratory treats biosecurity? If there is no credible candidate zoonotic origin theory, the most useful way to advance our thinking on the origins is to have one or more concrete and specific theories of how a lab origin might have occurred. Such a theory would ideally answer questions like, what kinds of experiments were being performed when the virus escaped? Was the release attributable to human error or mechanical failure? What virus genome was being modified when the virus escaped? In today's talk, which I've titled the Mojang Miners Passage Theory, I'll first introduce the most concrete theories of the origin of the virus suggested by other authors. For the most part, though, I will elaborate on what we consider the most promising explanation of the origin of the COVID-19 pandemic. It is a theory which I and my colleague Alison Wilson have contributed to, but in many ways it originates with a neglected Chinese master's thesis discovered by an anonymous tweeter called the Seeker 268. The theory also leans on a Chinese PhD thesis that the same seeker also found. The theory was then built on by two Indian researchers, Monali Rahalkar and Rahul Bahulkar, who later published a paper which is referred to in the further reading section of the last talk. Before we can discuss any of these theories, however, it is worth digressing a little into the virological technique known as passaging, because passaging features at some point in almost every lab origin theory. Imagine you are a researcher and you are given a cat infected with a mystery virus and you want to study it. If you extract the cat's virus and put it into a cat cell culture in a petri dish, the virus will presumably replicate in those cells and you can study its behavior, its mode of replication, its genetics, and so forth. Now, if you instead do what this slide shows and put the cat virus into a petri dish with human cells growing in it, what will happen? In all probability, the cat virus will replicate, that is, copy itself, but much more slowly. It may also spread between individual cells, but likely also poorly. When those cultured cells activate their antiviral defenses, which usually takes time to ramp up, the virus will likely die out and be lost. But an alternative outcome is possible. The cat virus may mutate and evolve to perform slightly better in those human cells. But even if some mutations do arise, the human cells will likely still eradicate the virus. However, if you as the researcher intervene and rescue the virus after it has acquired an adaptive mutation, but before it completely dies out, and then inoculate a fresh set of human cells with the rescued extract. Then the process will restart, but with the difference that this time round, the virus inoculum is partially adapted to human cells by virtue of any mutations acquired in the first round of infection. Imagine repeating this process numerous times. After not that long, the cat-adapted virus becomes a human-adapted virus by acquiring in a stepwise fashion a series of mutations that, for example, enable the virus to better suppress host cellular defenses or more tightly bind to human receptors. Perhaps this new virus can no longer infect cats or cat cells at all. You have created, in this fictional scenario, a novel human-adapted virus. This virus can then be studied to find out what are the differences between the cat version and the humanized version. However, there is a subtlety to passaging that is important to appreciate, as it is potentially key to understanding the origins of SARS-2. In our fictional example, the virus became steadily adapted to human cells. It did not necessarily become adapted to human bodies, however. The distinction between cell culture and whole organisms is very important. Human bodies have viral defenses that individual tissues and cell cultures do not. A successful virus must learn how to evade these system-level defenses in order to succeed at the organism and the population level. Let me give you an example. SARS-2, as many know, has a spike protein whose function is to bind to receptors, called ACE2 receptors, on the outer cell surface. In photos or models, the spike can look like a rigid structure, but in fact, it continuously moves. This ability to move appears to be a defense against the antibodies of the acquired immune system. The spike is an obvious target for antibodies, and this makes it a point of vulnerability. So one of its defenses is to be a moving target. The lesson here is that what constitutes a good adaptation in a cell culture, such as a spike constantly held ready to infect new cells, can be a hindrance for infecting a whole organism. That is to say, adaptation to a cell culture does not equate to adaptation to a whole organism. The requirements for adaptation to a whole body are always somewhat different, and they may, as in this case I just outlined, even be contradictory. The implication of this is that in order for the passaging of any virus to fully adapt to humans, this passaging pretty much has to occur in whole organisms. So that is passaging, also sometimes called serial passaging. It is the transferring of virus between cells or between organisms for the purpose of forcing its evolution or testing its evolutionary potential. Passaging is a fairly standard virological procedure, but it is also potentially a very dangerous one. Some of the most controversial experiments in all of the history of science have been virus passaging experiments, because, as in the fictional case I've just outlined, the result can be a virus adapted to a new species and thus the effective creation of a novel pathogen. Passaging is thus a technique for adapting viruses to other species, which is why it plays a role in most lab origin theories. The first published theory of a lab escape to appear in the peer-reviewed literature came out in the journal Bioessays, and the authors are Sirotkin and Sirotkin. Their contention is that the development of the SARS-2 virus may have been primarily a case of guided serial passaging. Some of the key points made in the paper are that serial passage mimics a natural zoonotic jump, and so is likely impossible post hoc to distinguish the two. They also believe that passaging, and I quote, offers explanations for SARS-CoV-2's distinctive spike protein region and its unexpectedly high affinity for angiotensin converting enzyme ACE2, end quote. This restates what I indicated to you in the previous talk. The fit of SARS-2 to humans is puzzlingly high for a recently evolved pathogen, and that feature is particularly well demonstrated by its binding to ACE2, the human cell receptor. In the last talk, I called the receptor ACE2, but its full name is angiotensin converting enzyme 2. Most coronaviruses only really infect the upper respiratory tract, yet SARS-2 infects and damages tissues such as lungs and nerves. Unlike any of its close relatives, SARS-2 also has a series of 4 amino acids whose side chains behave chemically as bases. The word basic in this concept refers to pH, as in acids versus bases. These amino acids make its spike susceptible to cutting by furin. Furin is a host enzyme that cuts proteins. The ability of its spike to be cut by furin dramatically expands the range of tissues SARS-2 can infect. Thus, the furin site alone may largely explain why SARS-2 is much more pathogenic than most coronaviruses. This furin site may also have enabled the species jump to humans. Therefore, a key question of researchers ever since the outbreak began has been to ask how this protein motif arose. Something the Sirotkin's point out is that passaging can give rise to polybasic furin cleavage sites. What the Sirotkins ultimately propose is that researchers at the Web, starting with their vast unpublished collection of SARS-like coronaviruses, could have created SARS-2 by passaging a precursor virus in primate cells or passaging it in whole animals such as ferrets or humanized mice, or some combination of the above. They also suggest that an accidental release into farm animals such as mink, or alternatively the cut and paste addition of a furin site in a lab, may also have played a role. In our opinion, these suggestions have merit and are plausible. In contrast to the Sirotkin paper, this publication by Rossana Segreto and Yuri Deigin focuses on mutagenesis and genetic manipulation as the method by which SARS-2 might have been made. They suggest that the purpose of this manipulation was either to make a vaccine or was part of experiments to test the evolutionary potential of novel coronaviruses or for drug testing. The authors focus on how natural or modified spike proteins and novel furin sites could have been added to make chimeric viruses without leaving any genetic trace. They point out that in 2017, for example, Zhengli Shi and colleagues at the Web published a paper describing chimeric viruses in which the spike protein from various SARS-related viruses they had discovered was substituted into the backbone of another SARS-related coronavirus called WIV1. In the engineering of SARS-2, a possible starting genome for such chimeras might have been RATG13, which is, as we know, the nearest known relative of SARS-2. This too seems a plausible theory. The third candidate lab origin theory that we think merits attention comes from a partly anonymous online group that goes by the acronym DRASTIC. In the DRASTIC theory, SARS-2 was an attenuated live vaccine that escaped. They nicely show, for example, that various unusual features of SARS-2 coincide with recent innovations in coronavirus vaccine research. This too seems realistically plausible. I encourage people to explore each of these theories, but to read Drastic's reasoning, you will need to follow the link provided at the end of this talk. So this is a reminder slide from the previous talk. I'm repeating it here to show the upper figure B again to remind everyone what coronavirus genomes look like. The lower figure C is more important to us though. It is a diagram called a genetic similarity plot. This one shows which viral genomes are most genetically related to SARS-2. The higher they are, the closer to identical with SARS-2 they are. The blue line, indicated with a blue arrow, is RATG13. Note that RATG13 is always the highest line. It is in fact twice as closely related to SARS-2 as any other virus known at the time that this figure was produced, which was January 2020. Overall, RATG13 is 96.2% identical to SARS-2. This means that RATG13 differs from SARS-2 by 3.8%, nearly 1,200 nucleotides. By ordinary standards of evolution, RATG13 is separated from SARS-2, therefore, by maybe 50 years of evolution. This, in many ways, is the crux of the issue of the origin of SARS-2. What explanation, zoonotic or laboratory in nature, can explain that gap? So, by the way, people familiar with this story will know that there is now another relevant contender, RMYNO2. But for various reasons, I believe it can be safely ignored for the purposes of this discussion. The virus RATG13 was discovered by Zhengli Shi of the WIV in 2013. The first scientific publication to mention it came out on February 3rd, 2020. This was the paper in which Zhengli Shi also presented the first ever SARS-2 viral genome sequence. The initial account of the long time gap between its discovery in 2013 and publication of RATG13's genome sequence in 2020 was that samples containing it had lain untouched in her freezer for 6 years until the COVID outbreak, when its potential significance as the closest related virus to SARS-2 became apparent. By the time we first began to look at the history of RATG13, online sleuthers had discovered that RATG13 had an interesting history. It was obtained from a mine shaft in Yunnan province, China, about 1,000 kilometers from Wuhan. These facts account for its name. RATG13 is an acronym. RA stands for Rhinolophus affinis, the bat species from which it was anally swabbed. TG stands for Tongguan, a town near to the mine. Mojiang, by the way, is the county where they are all found in, but it has become the English language nickname of the mine. The Mojiang mine is potentially important for the reason that in 2012, 6 miners had become sick, and 3 of them had died in a disease outbreak after shoveling bat guano there. Zhengli Shi had told Scientific American magazine in March that the mine outbreak was irrelevant to the discovery of RATG13, since the cause of the outbreak was a fungal infection. However, 2 Indian researchers, Monali Rahalkar and Rahul Bahulekar, had by then written a preprint referring to a Chinese master's thesis describing the hospitalization and treatment of the 6 Mojiang miners. They were treated at Kunming Medical University Hospital. Kunming is the capital of Yunnan province. The thesis is in Chinese, but its English title, Analysis of 6 Patients with Severe Pneumonia Caused by Unknown Viruses, was highly intriguing. The title was arresting, but the abstract was far more so. It implied that the miners' infections might have been a novel coronavirus caught while shoveling bat guano. The text is small, but I've underlined some of the references to bats, viruses, and coronaviruses, including the key word. It was hard not to notice that this is a very different story from the one given out by Zhengli Shi. We decided to translate the master's thesis into English. So I've lost my place. To assess the data ourselves. We also put the translated thesis online. The complete translation allowed a number of important conclusions to be reached. First and simplest was that the author of the thesis was the doctor who directly treated the patient. Second, the conclusions of the thesis were reached in consultation with eminent experts from across China, and the thesis provides notes or video conferences with them. The most significant and prominent of these experts was Professor Zhong Nanshan, who is arguably China's most famous scientist and who was the hero of the first SARS outbreak. These meetings helped to show that the conclusions were carefully reached in conclusion with world-ranking experts, but also that the outbreak reached the attention of authorities at the national level. These authorities included the Chinese CDC, whose head is George Gao. George Gao is now also famous thanks to the current pandemic, but we now know that his lab also sent a large team to sample the mind for bat viruses, including a student who wrote a PhD. This indicates that Gao and the Chinese CDC also believed the virus was the likely explanation. In other words, the conclusions of the thesis are backed by expert opinions. The third important conclusion is shown in this slide. It shows the thesis's summary of the symptoms of the patients in table form. We usually refer to these patients as patients 1 to 6. This summary gives a flavor of those symptoms, but the text shows in great detail timelines of exposure and disease progression, test results, CT scans, X-rays, and the treatments applied. The table highlights dry cough, high fevers, limb soreness, and in some patients, headaches. The text also mentions acute respiratory distress syndrome, commonly shortened to ARDS, and low blood oxygen, while CT scans showed ground glass opacities and bilateral lung infections. 3 minors were also treated with the blood thinners warfarin and heparin. The chief point to note here is how closely these symptoms resemble COVID-19. They are in fact clinically indistinguishable from it. For example, a classic observation for COVID patients has been excessive blood coagulation requiring treatment with blood thinners. But COVID patients typically also have high fevers, dry cough, and low oxygen levels and ARDS. Like the minors, patients often die in a state of high fever. In assessing the potential causative agent, the thesis ruled out many common and uncommon causes of the minors' infections. Importantly, they excluded SARS-1, since in principle the outbreak might have been a reemergence of that virus. Minors' blood samples were sent to the Wuhan Institute of Virology, and the descriptions of the test implies a test for coronaviruses, but is ultimately unclear. We subsequently learned from the thesis of George Gao's student that other tests sent to the WIV showed the minors had antibodies that reacted to SARS. The logical inference, since the minors did not have SARS-1, is that the minors had a distinct SARS-like coronavirus. In summary, what caused the outbreak in the mind was therefore probably a novel SARS-like coronavirus. A coronavirus infection also makes sense from what we know about the diseases that bats carry. Bats carry rabies and other viruses, but unlike coronaviruses, most are not associated with respiratory infections. Second, Zheng Lixi's subsequent sampling of the mind revealed an unusual abundance of coronaviruses. Most were alpha coronaviruses, i.e. coronaviruses, but not close relatives of SARS-1 or SARS-2. However, 9 of them were close relatives, including RATG13, which, as we know, is the closest relative to SARS-2. Hence, the conclusion of the thesis that the probable causative agent was a novel SARS-like coronavirus originating in a horseshoe bat has strong supporting evidence. The strange-sounding concluding phrase, bat kind SARS-like cough. By the way, is resolved if we consider that it refers to bats related to Rhinolophus affinis. Thus, the author is saying that the coronavirus is similar to SARS and it came from a bat species related to Rhinolophus affinis. Based on the totality of this evidence, in July 2020, we proposed a sequence of events that we called the Mojang miners passage theory. The theory is not published in the scientific literature, but it is published on our Independent Science News website. The relevant article is called A Proposed Origin for SARS-CoV-2 and the COVID-19 Pandemic. I mention this because there is no time in this talk to cite key references nor provide more than an outline of the theory and its supporting evidence. So please read the article. It is fully referenced. In an outline, the theory proposes that the SARS-2 virus derives from the body of one of the 6 miners, most likely patient 4. Our suggestion is that RATG13, or a closely related bat virus, infected the miners, and that simple evolutionary forces adapted this bat virus to human physiology, creating SARS-2. We suggest that when samples from the miners were unfrozen at the whip, they therefore contained a by then highly human adapted virus. It was this virus that infected a researcher, presumably in late 2019, triggering the pandemic. So this slide outlines key aspects of the Mojang miners passage theory. I will go through each point fairly briefly. The first point, we now know that Zhengli Shi sampled the mine for bat coronaviruses multiple times. Starting in July 2012, while the miners were still in hospital, continuing at least until 2015. These WIV researchers found unusually high numbers of coronaviruses. As well as RATG13, they found hundreds of diverse coronaviruses. 8 were close relatives of SARS-1, SARS-2, and RATG13. But how might a bat virus infect the miners when bat coronaviruses typically do not bind human receptors? Recently, researchers showed that the spike protein of RATG13 binds efficiently to the human ACE2 receptor. This provides a clear route for RATG13 to gain entry into the lung tissues of the miners. But there is another potential route of virus entry. We can reasonably suppose that the miners were inhaling dust and probably exerting themselves shoveling the guano. Particles of guano with virus attached could potentially penetrate their lung epithelium and enter some cells by purely mechanical means. It is consistent with the theory that RATG13 initiated infection alone. But given the unusually high diversity of viruses in the mine, an important consideration should be the possibility that the initial outbreak was caused largely by mixtures of viruses. After all, there ought to be something unusual about the mine that caused the outbreak, and the obvious candidates are a special bat virus, like RATG13, that happens to bind human cell receptors, or unusual quantities of virus, or the presence of multiple diverse viruses. All of these potential explanations are in play, given what we know of the viruses in the mine from the subsequent sampling. Let us then look at what happens when more than one virus infects a tissue at the same time. Let us assume that this virus mixture is composed of viruses, each with varying levels of competence to replicate and spread in human lung cells. This is a cartoon depiction of lung epithelial cells with different coronaviruses inside them and an air passage above. I've shown the genome and the envelope and the spike of these viruses. I've drawn 4 spikes per virus, but the usual number is closer to 40. In the lower left can be seen a green virus that, let us assume, can replicate in a human cell but not spread. Perhaps its spike is incompatible with human ACE2. In the middle is a plum virus, which can hardly replicate or not replicate at all when it enters the cell. On the right, in red, is a RATG13-like virus that can replicate and also spread. Obviously, the red virus is a threat to the miners. But what about the plum and the green ones? Here, as expected, the red virus is spreading to neighboring cells. The green virus is replicating but not spreading, and the plum virus is doing not much of anything. Now what happens when the red virus enters a cell with either the plum virus or the green virus? In the case of the plum virus, the red virus causes it to replicate because it supplies the lacking replication functions. On the other hand, the red virus causes the green virus to spread. It does this because when they share a cell, the spike protein of the red virus becomes incorporated into the envelope of the green virus. The technical word in biology for this phenomenon is complementation. Complementation occurs when one organism supplies functions that another one lacks and so overcomes its defect. If we follow the process a little further, we see that the purple virus is now spreading and the green virus is too. But note also that their spread is dependent on the presence of the red virus in the same cell. I've given this example emphasizing specifically the sharing of spike proteins and replication functions. But in principle, whenever two different viruses infect the same cell, there is the potential for the viruses to complement each other regardless of the function concerned. Soon enough, though, another thing is going to happen: genetic recombination. The purple virus is going to acquire the genetic part of the red virus that enables replication. And the green virus is going to acquire the code for the spike protein from the red virus. The green and purple viruses will then be independent of the red virus and can spread on their own. How often is such recombination thought to occur? The evidence for coronaviruses is that if a researcher places two distinct viruses in the same cell, by the time the virus has finished replicating, 10% of all the progeny will be recombinant. In other words, coronavirus recombination happens straight away and continuously. Coronaviruses recombine at very high rates. This goes unnoticed in normal infections because the population is composed of virtually identical viruses. Likewise, in a lab setting where research is done mostly with pure strains. What we are suggesting, therefore, is that within 24 hours of the initial infection of the minus lungs, all of these events will likely have occurred multiple times. And if they occur in different parts of the lungs, each infection focus would begin with slightly different starting viruses and evolve in slightly different ways. So without even discussing the added evolutionary possibilities of simple genetic mutations, a situation is occurring that represents a very interesting breeding ground for the potential future evolution of the viruses in each miner's lungs. Very rapidly, viruses would likely have arisen in the miner's lungs that differ substantially at the sequence level from those originally present in the mine. This analysis of the early stages of infection in the miner's lungs is one way to answer the big question I mentioned earlier, and which the Mojang miners' passage theory needs to solve. How could 50 years of evolution from RATG13 to SARS-2 happen in one miner's lung? This is the main objection in our experience that has been raised by proponents of other origin theories. So the next few points are also intended to support the case that it can. The general argument is that standard estimates of coronavirus evolution are derived from measuring a very narrow set of conditions. These conditions largely exclude mixed infections, recombination, and species jumps. For example, whose typical effect is to radically accelerate viral evolution. The third point I therefore want to raise is the lengthy hospitalization of the miners. The miner's thesis provides a timeline extending from initial exposure in the mine to discharge or death. From it, we can see that patients 3 and 4 were hospitalized for 109 and 107 days, having been first exposed 23 days earlier. When we propose that RATG13 evolved into SARS-2 more or less entirely inside one of these miners, the relevant timeline is the date of first working in the mine, April 4th, to the last tissue or patient sampling, which in the case of the WIV was in September. Going further towards answering the question of how so much genetic change can occur, there are likely to be key differences between the situation occurring in the miner's lungs and an ordinary transient coronavirus infection of the upper respiratory tract. The first difference is that lungs represent a much larger tissue than the upper respiratory tract, approximately 1,000-fold greater, several kilos versus a few grams. In other words, the miners supported a far larger virus population than a normal human coronavirus host, allowing proportionately greater evolutionary change. We also suggest that recombination played a significant role in later stages of infection. After the initial period, the processes outlined above would have resulted in infection foci dispersed through the lungs. Certainly, distinct foci is what the CT scans of the miners show. These foci would likely have represented distinct lineages of viruses. Because the lungs are connected by airways, these lineages would nevertheless likely be able to exchange genetic material, which would also speed their evolution. The third possibility worth consideration is that the treatments and diagnostic tools applied to the miners might have affected the mutation rate of the virus. CT scans and chest x-rays, for example, are mutagenic and were given many times to the miners. But how this affects RNA is not well understudied. We suggest that all these phenomena, but especially the distinct infection foci in the lungs, would have contributed to the creation of passaging type conditions in each miner's lung, which is why we call it a passaging theory. From various sources, we know that samples were taken from the miners for analysis, and some were sent to distant institutions. Throat swabs and blood samples in the thymus of patient 4 were taken, and some or all were sent to the Chengdu military region for disease and control. Also, Dr. Zhongnen Shan in Guangdong, and to the Wuhan Institute of Virology. In a recent important development, in the November addendum to her original February paper in Nature, announcing the publication of the first genome sequences of SARS-2 and also of RATG13, Zhengli Shi acknowledged for the first time any positive interest at least, in the miners, and also to having received 13 blood samples taken from the miners. They came from Kunming Hospital in 2012. There is therefore no longer serious doubt about a key step in the theory. The samples from the miners arrived in Wuhan. I've outlined the case that these samples may have contained SARS-2. According to Zhengli Shi's addendum, however, these samples tested negative in PCR and antibody tests. But as I have already noted, the 2016 PhD of Kanping Huang, George Gauss' student, asserts that minor blood samples sent to the WIV were positive for SARS coronavirus, though the 2013 master's thesis is more ambiguous. But if one passes her addendum carefully, it is not clear that the tests she is describing are the same tests described by the thesis. There may be no contradiction. What is clear, however, is that the samples from the miners ought to have seemed extraordinarily valuable. as someone whose research career is predicting that coronaviruses can spill over into humans, the story of the miners ought to be of compelling interest. If a spillover into the miners was an epidemic averted, then the miner samples would have been proof of that theory and a compelling vindication. Consequently, the silence of Zheng Lishi over the miners until the November addendum was essentially forced from her constitutes a grand mystery in the context of the current pandemic. Was she covering up an undisclosed research program at her institute and now an accident? Or is there an innocent explanation? I've more or less run out of time for this week, but I would like to end by saying that whichever explanation is the true one, Zheng Lishi is not the only person with potential inside information concerning the web who appears to be protecting it. So in next week's talk, I'll report on some of our investigations into the conflicts of interest that are behind the failure to properly investigate the origins of the virus, and that can explain many aspects of the pandemic response as a whole. So here's some further reading for you. So thank you very much, and I'll hand over to whoever is comparing this, and maybe we can have some questions. So I'm happy to take whatever people want to ask. So thank you. Can you repeat that question, please? What further questions do I have? Yeah, so that's a good question. I mean, it raises, what the addendum does is it talks about the provenance of RATG13, and it also describes what happened with samples that were brought from the Kunming hospital. So these are pretty different questions, but what is in common with all of the answers given by Dr. Shi is that, they're not, basically the addendum is very short, right? It doesn't provide, it provides theoretical answers, but it basically says to trust her. And, given that she's changed her story on multiple occasions, it's difficult to do that. So what you would like to see is, more accurate descriptions of what actually the tests were that were done. So she describes antibody tests that they did that she claims rule out the possibility that the pandemic, or she implies rule out the possibility that SARS-2 was in the minus serum, but she doesn't, for example, describe experiments that correspond particularly well to the ones described by the students, and she doesn't describe the experiments that she actually did. Like, it would be nice, for example, if the addendum went through a peer review process and we could see the actual evidence of the experiments that she did. For example, what antibody did she use? What dilution did she use it at? What did the blots look like? What were the controls? And so, the standard things that people offer when they describe experiments. These are out, it's treated as if, we should just believe whatever word it is that Dr. Shi has to say, when these are outstandingly important experiments, right? We're talking about the basic origins of where this enormous pandemic disaster has come from. And we are basically expected to take the word of someone that there was no pandemic virus in these samples. And so this is very problematic. And the addendum, for example, should have gone through a peer-reviewed process. And I don't think it did that. I think Suzanne is talking about the pandemic preparedness kind of workshop that was organized. I believe it was paid for by the Gates Foundation. I have not seen, I haven't watched it. I believe one can watch it online. And, what to me is interesting about all this is there's not a surprising thing. We've had near misses from pandemics for multiple times, right? We've had MERS, and we've had swine flus and bird flus and SARS and so on, Ebola virus. We've had all these near misses. And what's interesting is, the health system has kind of leaned in the direction of, we'll solve these problems with high-tech approaches, like vaccines that, we don't have already, or through, complicated diagnostics, or through development of, patented drugs. But there are very simple ways of preventing pandemics becoming disasters. If people are provided with more masks, if hospitals are provided with plenty of PPE equipment, if people are, well nourished and looked after, there's a whole ton of just kind of general preventive measures that could be done that would have many benefits, that wouldn't rely on precise identification of what is the pathogen and where it comes from, and what how developing a diagnostic kit, for example. It took quite a while for a diagnostic kit to come, but you don't need, if you store masks, for example, then you don't necessarily need all that, because you have ways of protecting the population that don't rely on patented and high-tech solutions. But what's interesting about the preparations that we've chosen as societies, England and Germany and the US, pretty much every country have chosen to rely on high-tech damage response type answers to the problems posed by pandemics. And I think that, this is one of the lessons that to me is a take-home thing. We shouldn't be relying on those things, or at least we shouldn't be relying on them solely. We still have hospitals rationing masks, and not enough masks, and not enough information about masks, all these questions that, show a kind of mismatch, in my opinion, between official thinking about how to deal with pandemics, and it was not solved by this gaming event. And so I see that these gaming events are not necessarily very useful. And so that's a question that should be asked too. The Gates Foundation and some others have put themselves in the position of trusted advisors and so forth to governments in terms of how to deal with the pandemic. but the evidence, my reading of the evidence is that they have not shown themselves to be very competent. Mm-hmm. Well, if I understand the question right, Benjamin is asking about whether the scientific community is discussing this event properly in terms of looking at the origins. And it seems very clear to us that people are protecting the with, whether or not they understand what happened there. I would say that we don't know fully what happened there, but there are lots of people who don't know any better than we do what happened there, even though many of them are much closer to the real events. But they are kind of in a knee-jerk way protecting the scientists close to this system. And so what you have is basically people who are scientists, but who feel that they can't discuss these issues in public or in the open without being kind of accused of disloyalty or conspiracy theories or being, some kind of hysterical person. And what's happened is that people at the, closest to this thing have done quite a good job of, it seems to me, deliberately politicizing this issue. When the Lancet letter came out, for example, there's a Lancet letter in, I think it was March. It came out and it basically said that it was signed by 27, well-known pathogen biologists. And it basically said that anybody who talks about a lab origin is a conspiracy theorist. But we know, and I outlined some of the evidence in the previous talk, that there are plenty of examples of lab-escaped viruses, let alone other pathogens. And so we have lab-escaped pathogens that have become pandemics. We've had lab-escaped pathogens that have been near-miss pandemics. We've had lab-escaped pathogens that were not discovered at the time. We have an extensive history of lab escapes from many different countries with many different viruses with many different situations. There's nothing ridiculous about proposing a lab escape. There's nothing conspiratorial. So when people put pressure on scientists not to talk about this, then it creates a problem for society. And so this is why people are investigating the origins of the virus and posting things online, basically anonymously, like Drastic, for example. But there are others. There's a group called Heraclitus that posted some articles online about the origins of the virus, and they refer to ours and some of the other ones I've discussed today. But they basically said we're doctors and professionals who would like to be anonymous because there may be professional repercussions, right? Well, this is not a satisfactory situation, and if we think we're a free society, then we should allow people to discuss things as they see fit, right? So there's a whole mismatch between the professed, status of science as being an open place of discussion and the reality that people feel subtle and not so subtle pressures to keep quiet about what they think are the origins of the virus. And what that means is you don't have a proper discussion of it in the scientific literature. There's very, very few, and I've brought many of them to these talks, but there's hardly any scientific publications that have actually discussed the origins of the virus in a way that encompasses lab origins as a serious hypothesis. And people feel, it's pretty obvious that scientists feel intimidated and frightened from speaking out. And so the people who are speaking out for the most part are people who are, ex-virologists or near virologists or very interested members of the public and so forth. So we have a democratic problem of scientific integrity, basically. So yeah, it's a huge problem. You know, this is not a trivial issue, right? Whether it came from a lab is a question that basically will determine the future outcomes and how we respond to pandemics. You know, will we continue to do the kind of research that potentially that led to this pandemic? You know, if it happened at the Wuhan, let's think about this. If it happened at the Wuhan Institute of Virology, we can theorize that it might have been a bioweapon or something, right? But the more likely thing, in my opinion, is that it's simply a lab accident. But then the question becomes, what kind of experiments were being done in this lab accident? Were they passaging the virus? Were they making a vaccine? Were they doing experiments on drugs? Was it an experimental challenge molecule? Did it result from, you know, the reckless collection of coronaviruses from the wild? We can learn a tremendous amount if we understand what happened. But if we don't understand what happened and everybody covers over the possibility that it was a lab origin, then we're going to go, you know, close down animal markets and stop people hunting wild animals and do all kinds of, put in place all kinds of responses that arguably are completely not evidence-driven and inappropriate. And the consequence will be that we don't learn from the pandemic and it happens again. And is that what we want? Obviously, it's not. No, it does not. Yeah, I can add something to the previous question too. So you know, the Conrad, I think, if I understood that name right, asked whether RATG13 has a furin cleavage site, and it doesn't, which means that the cleavage, you know, if our theory is correct, that the cleavage site must have evolved inside the minus. And what we proposed actually in our, you know, it's simply a suggestion, but what's been observed is that the humans are the only organism that have a furin cleavage site the same as that supplied by that evolved in SARS-2. And our suggestion is that it evolved by recombination with the RNA of the protein that contains that furin site, which we happen to know is present in abundance in human lungs. So our theory is that basically the virus obtained the furin site from recombination with a human RNA while it was inside the minus lung. So I just wanted to add a little bit tonight because I gave a rather short answer to your perfectly good question. So, you know, the question from Fabian is very interesting. You know, is there some kind of other explanation that could be brought forward that, you know, basically implies why people would be defensive and why people would cover something up, but doesn't involve a lab origin and a lab escape and some kind of, you know, nefarious or secret or whatever they were activities on the part of Zheng Lishi and the whip. And I think that, I think it's a good question to raise because, of course, the whip could have hired these miners to collect things and be, kind of embarrassed about what happened. But I'm not sure that that kind of embarrassment could have explained the kind of misinformation that, I'm going to talk a little bit about this next week, but, going back a very long time, and this would be consistent with Fabian's suggestion, the defensiveness of the whip and Zheng Li appears to go back a long time. So in their first sampling paper from the mine, they title the paper that and the samples come from an abandoned mine, right? Which, seeing as we now know that there were miners digging guano, supposedly in order to in the future mine copper, it's inappropriate to call that a abandoned mine, because at the time all of this was conceived, it wasn't abandoned, and the only reason it's probably abandoned today is because the Chinese authorities have banned people from going there because they might start a pandemic, right? So that's a strange thing. Like, it doesn't even belong properly in the title of the paper to call this mine an abandoned mine. So, and that paper also does not mention the miners or their illness, right? As I said, this would be incredible opportunity for Dr. Zheng Li to publish, famous papers about an averted coronavirus epidemic and to show what the scientific basis of human adaptation of viruses inside the bodies of these miners. The scientific possibilities for her are almost infinite. And yet, even though she doesn't know what's going on with these miners, she's already saying that the mine, in effect, by not mentioning them at all, that she thinks that they're irrelevant to this outbreak. So this is, I don't think you can build a case around that particular scenario, but I think these are questions that have to be asked, alongside all the ones that I've put forward.
Hi, John.
Hi there.
How you doing?
Hey,
actually,
for coming to this.
Actually, I'm writing a book about COVID-19, and I'm just finishing a chapter called "The Origins of COVID," and I quoted a lot of you and Yuri Dagin and some other people. So I'm curious about the two crucial things. Are the relationship to the furin cleavage site and then the spike protein itself,
Yeah.
and the RBM.
Yeah.
So, right. So the thing is, it's a polybasic furin cleavage site, and most people say the mare's has a furin cleavage site, but it wouldn't be able to be passed on to the SARS-CoV-2 because it's in a different family. And the question is it a polybasic cleavage site in mares? In other words, is it more likely it's synthetic, or is there what organisms, what coronaviruses have a polybasic furin site?
Yeah, I mean, all furin sites, to my understanding, are polybasic somehow, but they achieve that basic, you can rearrange the basic amino acids and so forth and still have a polybasic furin site. And I don't recall directly the MERS site. I've looked at those little alignments and so forth in the past, but I can't recall offhand what is the nature of that site. But it's not true that it couldn't recombine. Viruses recombine with homologous and non-homologous sequences. So in principle, there is no reason why that it could not, if it happened to be, if the two ancestor viruses were present in the same cell, they could in principle have acquired it from each other. It's not necessarily true that viruses only recombine with close relatives. There are classic examples from the scientific literature of radically different viruses that have recombined together. So we know that that's not necessarily a limitation. I have a little quibble with your premise about the emphasis given to furin cleavage sites and to spike RBMs, and spikes and RBMs, really. So RBM, for people who don't know, is a part of the spike that actually physically binds to the cell receptor, the ACE2, right? So there's the RBM is a part of the spike, a small part of the spike. So, my personal view of these viruses is that the whole virus has to be adapted to humans, right? We as biologists, tend to reductionistically focus on the small parts that historically people have focused on. And, we have our reasons, right? The exact domain that binds to the spike and the exact domain that's caught with the furin site. But every part of a virus protein and every molecule virtually of its genome has a purpose. And those purposes are suited to the host that they're in, and they're suited to interactions that go on within the virus itself. So, people focus very heavily on the furin cleavage site and the spike RBM. But to us, to our way of thinking, the mutations in the virus could arise at any point in the whole genome and cause the virus to be either better adapted or worse adapted to people and humans, in the course of their evolution. And I'm uncomfortable with people ignoring all the rest of the virus. And there are reasons why people focus on these certain parts, and they're certainly, there is a reason, but there's also a reason to focus on the rest of it too. And so I don't give them the primacy that most people do, which doesn't mean they're not important. It just means sometimes that they're not so variable, or that they have a biochemical logic that appeals to grant writers and funding bodies. Does that answer your question properly?
Yeah, that's very good. In other words, the old Montaigne statement, the more we know, the more we know we don't know.
Yeah.
Right? You have all those different amino acids in combination, and who knows what they do.
Yeah.
That's what you're saying.
Yeah, viruses are infinitely complicated. I used to work on viruses that had overlapping open reading frames. So you'd have proteins, in a sense, hidden inside other proteins. And every part of all these viruses, every tiny little motif has some, place where it interacts with the human genome or with its host, not so much with our human genome, but the cell functions. So the plant virus protein that I did some research on a few years ago, it was a tiny protein. It had 7 different functions. And those are ones that were just known about. And if one was to research, if there were more than 2 or 3 labs working on that protein, I bet you'd find a whole bunch more functions for that protein. But we don't know what they are because we just don't do enough research. We don't even know how to work out, how these viruses interfere with their host cells and so forth. So there's a whole, we don't give credit to what we don't know enough. So I'm absolutely with the Montaigne statement. Yeah.
Thank you.
Hi, it's a little noisy here. I missed part of that conversation, but I was wondering, is it possible that this was done on purpose?
Sure, that's a perfectly reasonable question. What's interesting is that there are some aspects of the virus that are a little hard to explain by accident. You know, if Zhengli Shi is hiding information going back a long time, what that implies is that their concerns about people finding out what was being done predate the pandemic, right? If they have for example, you know, not referring to viruses that they took from the mine. For example, why would no one talk about the viruses that we now believe came from the mine? There's a lot of strange things going on here. And so we have to keep all our options open. I mean, I personally don't think that is the best explanation. I'm pretty loath to put forward the idea that that's what was going on. But it is, you know, it is consistent with certain parts of the data that there's more going on than just this pandemic. And you know, the one thing that's in favor of it in part is that people say, well, it would be a terrible bioweapon, for example. If somebody wanted to unleash a bioweapon, why would you use this one? Hardly kills anyone. But the answer in part to that is that, you know, the history of these SARS-related coronaviruses, a little narrow group that the study that the WIV was interested in, is that SARS-1 was a member of that. SARS-1 kills 10% of the people who are infected with it, right? And these numbers, you have to take these numbers with a grain of salt because they're all about, you know, the health of the population and how well you know how to treat people with these viruses. You know, these are not absolute numbers, but the SARS-1 was a very bad virus. The virus, you know, if it was a virus that was killing these miners, it was killing 50% of the people, right? So there'd be reasons to think that what was in the miners would be a very harmful virus. So the fact that what's released is not particularly harmful, it's not good, but it's not bioweapon standard virus, that is not necessarily the right argument to be using. So I don't want to close off that option because you know, that it remains a possibility, and I don't think that anything can be taken off the table at this point.
Hi, Jonathan. Thank you so much for this great presentation. It really helps me to listen to you talk about this after reading your excellent paper. So I have a question about the Mojiang miners and whether they actually are from Mojiang. I know I've asked you this before via email, and you've said yes, Mojiang is mentioned in the thesis, but it's only mentioned once, referring to one paper, and Mojiang is 250 kilometers away from Kunming. And the reason why I wanted to ask this is because coincidentally, Shi Zhengli and Peter Daszak were collecting bat viruses from, quote, a single location in Kunming between April 2011 and September 2012. And it was on this virus hunt that they found a bat SARS-like coronavirus that uses the ACE2 receptor. And this was RSSHC014. And that's the virus that Ralph Barrick then used in his infamous gain-of-function experiment. So the virus hunt overlaps with the months that the men were in the hospital, which is April and May of 2012 in Kunming. Don't you suppose you could even just investigate this?
I mean, how would you do that? I mean, you know, we don't have access to the records of these miners. I mean, a little bit of information is contained in the master's thesis, and I seem to remember that there's some information about where the miners went home to, and that it wasn't particularly local, or where one of them went home to. But you know, my memory of this is quite dim. I mean, do you have in mind a scenario of what might have happened?
Well, you've been talking about why would Shi Zhengli and Peter Daszak want to hide everything about this incident. And you mentioned that as a possibility, it's possible that they sent people into a cave to collect guano. And it was in one of these virus hunting expeditions where they use very little personal protective equipment. They're getting bitten by bats. They're they're not really behaving like they're collecting deadly viruses, even though they are supposedly hunting for SARS. And so it is possible that it was the Kunming virus expedition where these guys got sick. And then, of course, what you're saying, you know, the whole thesis of your paper is that they collected samples from the miners, not from the bats necessarily. Like, of course, they collected samples from the bats, but they also had samples from the miners, and they had viruses from those samples potentially. And they were potentially doing gain-of-function experiments on viruses collected from human beings. And that's another reason why they might want to hide the specifics of what happened. But then there's RATG13 itself, which is marked in the Genome Sequence Archive as extracted from bronchoalveolar lavage fluid. And, you know, everybody says that's just a mistake, that it was, you know, in other places it's marked as a bat fecal swab. But these are just too many mistakes and coincidences, I think, for there not to be a full investigation. And I guess my last thought on this is that we are totally focused on the Wuhan Institute of Virology because the virus, COVID-19, the pandemic starts in Wuhan. But it's entirely possible. I mean, we know that Ralph Barrick had at least one of these viruses. Xi Zhengli's partner is Peter Daszak with EcoHealth Alliance. He's funded by the US military in addition to other US agencies. There's a lot going on here that I think deserves an investigation.
Yeah, I totally agree. I mean, yeah, it's hard to see such investigations happening, but you know, you've opened, you know, a can, you know, several cans of interesting worms, right, with your questions. And, you know, there is obviously, if somebody was collecting viruses from humans, there were especially humans that they put in the way of harm, that would be an ethical, you know, ethically, it's hard to explain that away, right? So that would be a reason. I totally agree. That is potentially a reason why one might not want to tell people about what you've been doing. You know, the other issue of the provenance, the sequencing of RATG13 is really interesting because, as you probably know, there's question marks about the sequence itself, right? I mean, right from the beginning, when I saw that sequence, it's very clear that this is not a viable, probably not a viable virus, right? I skimmed over all these things, but you know, the tradition in virology is that a virus is not a real virus until you sort of more or less fulfilled Koch's postulates, right? You've shown that you can take it out of the organism, that you can put it back into the organism. It causes a disease that you took it out, you know, you're seeking to explain and so forth, right? You have all these series of experiments and you have a defined strain that you say causes this. And they don't have, for RATG13, any RATG13, any of that kind of information. And that means that just sequencing with the contents of a tube does not provide you with a definitive virus sequence. It may not even be a functional virus. I know that people have been trying to make RATG13 in a lab and infect cells with it, and we've heard nothing about the results, right? And it may be that it's a totally not viable virus, and that the sequence that they've obtained is, you know, irregular, you know, defective in all kinds of ways. And that was one of the interesting things about the Anderson paper to me, is that they built their kind of defense of it's not an animal, it's not a lab origin a scenario that we're talking about here, based on the sequence that they must have known was not very authentic, because you've got, apart from anything, the seeming concept that basically this whole virus has been sequenced within a couple of weeks, right? So you know that this is not a virus that anybody has really managed to pass really carefully and do careful work on. You kind of hope that what you offer is right, but it's probably not. And then we also know about RATG13 is that if you do deep sequencing on it, it looks like it didn't come from a bat's bottom, right? The bacterial sequences that are in there don't correspond very well. And so, and like you say, there are records that the Shi lab has supplied, which imply that the virus didn't come from bat either, right? They come from bronchiolar lavage, which presumably is something that one does on a person, right? You certainly can't do it on a bat. So that raises all these really interesting issues, like is RATG13 some kind of virus that was offered up as basically a way of covering up what really happened? And this is something that occurred to us quite early and has only become stronger. When the first set of papers basically offered sequences of the virus, the SARS-2 virus, what happened was a whole succession of Chinese labs brought out these sequences, and Zhengli Shi got to go first, even though she doesn't seem to have been particularly on the ball. And she got to go first, and the others who published at the same time basically didn't have RATG13, right, available to them. So all they could conclude was that the nearest known relative of RATG13 was in the freezers of the Wuhan Institute of Virology. So this is the sequence 4991, BTCOV 4991, which is now considered to be identical to RATG13, but I think that's questionable. So basically, you can imagine a scenario, right, at the very beginning of the pandemic, in which everybody is, looking at these sequences, and they're all saying, there's this new virus, and it doesn't really have any close relatives, but the one that it does have is basically in the freezers of the Wuhan Institute of Virology. And if you think of that, this must have set the rumor mill going, right? Big time. Because if the nearest known relative is in the Wuhan Institute of Virology, and the virus came from Wuhan, you can't really reach any other conclusion that it was a lab accident. And so what happens is Zhengli Shi gets to go first, publishing in the scientific literature her sequence of the virus, and at the same time she publishes this seemingly cobbled together, or one suspects cobbled together, essentially that it's cobbled together, virus RATG13. And what that basically does is that locates the origins of the virus in a place that no longer implicates the institute, because you're basically saying, well, we never did anything with this virus. We had to pull it out of the sequence, pull it out of the freezer last week, because we find that it's in, based on a little fragment that we already had, we know that this is a close relative, and therefore it's super interesting. So what that does is deflect from the fact that they had the real original virus in their freezers. And this may be the reason why there was all this jockeying for position there's an article by Associated Press that's really interesting about all the labs at the beginning of the pandemic who sequenced the virus but were not allowed to publish it. And this is a public health problem because everybody wants to know what's the sequence of the new virus and no one's allowed to publish it. And then Zhengli Shi gets to publish first, right? And our explanation of that is that she was getting RATG13 ready so that she could cover up the tracks of the Wuhan Institute of Virology. So there's one explanation of what was going on there. Because the other piece of data to support that is that George Gao published a paper, was one of the publishers of papers that came out exactly at that time. And their paper made no reference to the virus that was kept in they couldn't reference RITG13 because they didn't know about it, but they made no reference to the virus that was in the freezers of the Wuhan Institute of Virology. So these are, renowned sequence experts and phylogenists, and there's a virus that's perfectly closely related to the pandemic virus, and they failed to mention it in their paper. Right? This is pretty interesting observation, right? Because it implies that they were in on what was going on with this virus. And then there's a third paper that's published at the same time, which does refer to the paper that's in the freezer, the virus fragment that's in the freezer of the Wuhan Institute of Virology. This is the 4991 sequence. And these are presumably people who are not in the loop, right? Who don't understand that you're not supposed to mention this virus. So that is our guess as to what happened early on. And then the last thing that doesn't add up about RATG13 and that we now know is, and you probably know this, Alexis, I'm sure you do, that the sequencing of RATG13 was largely done in 2017 and 2018. Right, so Zheng Lishi is claiming up till November that this is a virus that they never touched, and that they just sequenced it in the few weeks between the pandemic starting and then obtaining the sequence of the virus and publishing a paper in Nature in February. Right, they rush out this sequence with the implication, they don't say really where it comes from or when they sequenced it and so forth, but totally create the impression that they just whipped it out of the freezer and sequenced it that week. And that's not true, right? They were working on RATG13 sequence for years before that, dating back to 2017. So the question is, what were they doing with that sequence? Why are they sequencing part of the virus? Was it part of the research project that that I suggested they might have been covering up for? Because they never published that sequencing. So we don't know where what the scientific purpose of that was. But, it's an interesting virus. There's no reason why a person shouldn't publish it. And it's very closely related to SARS, for example. So it has many interests, that virus. And yet they were working on it and they didn't tell anyone. And seemingly they tried to cover up the fact that they weren't working on it. So multiple questions about this that, they do a little bit make one concerned about bioweapon issues. And the very last thing I wanted to pull out from what you said, Alexis, was that, it's also true that samples from these miners are being sent to other places. Dr. Zhong Nanshan has samples, right? Serum samples and so forth, right? So there are samples all over China that could actually demonstrate the truth of this theory, right? If you could just obtain viruses from samples that were extracted from the miners, we could show in a week whether this theory was likely to be true or not. So there's information that ought to be out there that, even though China is supposedly looking everywhere for a virus, there are named sequences in freezers that could tell us the answer. No, I would just like to thank everyone for attending and encourage you to, excuse me, to share these videos. That would be great. Thank you if you like them.